Hi everyone, my name is Lansing, and I am a bioinformatician working for Agriculture and Agri-Food Canada. Today I'm happy to be presenting some of the work that we've done on characterizing the resistome of honeybee gut microbiome via shotgun metagenomic sequencing. Here's an overview of the presentation. I'll begin with some background on antimicrobial resistance, as well as the importance of honeybees and their microbiome and resistome. I'll follow that with some technologies, methods, and samples that we've used in our analyses. And following that, I'll talk about our results. First, for the background. I'll start by defining antimicrobial resistance, or AMR. Antimicrobials are used to prevent and treat infections from bacteria, fungi, viruses, and parasites. When the target of the antimicrobial develops resistance to the treatments, this is considered antimicrobial resistance. The collection of AMR genes, or ARGs, involved in that resistance is called the resistome. Misuse and overuse of antimicrobials has made AMR a growing concern in the fields of medicine and agriculture, including apiculture, the management of honeybees for honey production and crop pollination. While everyone enjoys honey, the use of honeybees in crop pollination has enormous importance. Based on 2016 harvest data, the most recent estimate of the economic contribution of Canadian honeybee pollination determined that between $4 and $5.5 billion in additional crop values is made possible through the pollination services provided by beekeepers and their honeybees. Additionally, an estimate of over 80% of crop types are pollinated at least partially by insects. This provided a striking visual when a Whole Foods grocery store removed all the insect-pollinated produce, as well as other insect-pollinated favorites, such as almonds and chocolate. The honeybee gut harbors a host-adapted set of 5 to 9 core bacterial species clusters that occupy particular niches and locations along and cross-section to the digestive tract. These clusters can have substantial strain-level diversity. Transmission routes of the microbiota are primarily through social contact, similar to those in mammals. Most of the members are fermentative, breaking down the carbohydrate-rich diet of the honeybee, and have been subsequently hypothesized to contribute to host nutrition. A normal microbiota has also been associated with lower levels of pathogens and infection, potentially indicating a role of the microbiota in immune protection. An example of the importance of antimicrobial resistance in honeybees is the pathogenic bacteria Paenibacillus larvae, which causes the widespread and fatal disease American foul brood, or AFB, infecting larvae and reducing them to ropey slime, as seen in this image. In Canada and the USA, AFB has been treated with the antibiotic oxytetracycline for years, and since then, AFB with resistance to oxytetracycline has become common. Resistant AFB has subsequently been treated with the macrolide tylosin, but recently tylosin-resistant strains of AFB have also been discovered. The monitoring of the resistome could allow for the tracking of AMR strains and the informing of treatment method. It could also allow for the identification of reservoirs of ARGs that could transmit resistance to pathogens via horizontal gene transfer, as Okamoto et al. Investigated in their 2021 paper on AMR in the bacteria found in honey. Seeing as they disperse and collect water and nutrients, honeybees have also been investigated as potential vectors and monitors of environmental AMR. Honeybee resistomes have the potential to be used as a proxy for geographically distinct ARGs, which is exemplified in the Sun et al. Paper that came out this May, in which honeybee resistomes from across China were examined. Technologies and methods. In most of the literature regarding the honeybee resistome, microbial members have been isolated and cultured for sequencing. What we have done is used whole metagenome shotgun sequencing for resistome characterization. I'd like to draw some comparisons between these methods. Bacterial isolation and whole genome sequencing can be useful for producing strain and plasmid level sequence definition, both of which can be important when it comes to the detection of ARGs. So what are the benefits of resistome characterization via shotgun metagenomics? Well, the results are at the resistome level rather than individual genomes, providing a wider context. As well, in the project from which our samples came, there are hundreds of samples from various experiments, all sequenced with the wider purpose of metagenomics analysis. The methods of in silico resistome analysis that we've used can easily be performed in addition to other metagenomics analysis, such as metagenomic assembly, which we've also utilized. Additionally, these methods are easily scalable to a large number of samples in comparison to bacterial isolation. Our samples were taken from honeybees in Ontario, Canada in 2020. From 10 separate sites, 3 bees each were taken from 4 colonies. For each sample, those 12 bees had their guts removed, pooled, and sequenced with Illumina shotgun sequencing, producing an average of 68.8 million base pairs per sample. These sequence reads were processed through the AMR++ pipeline, producing short-read resistome results classified against the MegaRes ARG database. The same reads were also assembled into contigs with metaspades, and these contigs were screened for ARGs against 5 ARG databases, including MegaRes, using the tool Abricate. A little bit of information about AMR++. This pipeline performs both taxonomic classification as well as resistome analysis after quality control and host filtering steps. Today I will be sharing only the resistome analysis results. AMR++ identifies ARG sequences by aligning them to the MegaRes ARG database, followed by Resistome Analyzer, which deduplicates and minimizes the potential for false positive classification by removing sparse alignments. There is an additional step that searches for particular SNPs in genes that may otherwise be wild-type, non-AMR genes. Genes that require SNP confirmation are typically those with other primary functions that gain AMR through mutation. We've excluded these genes, and thus this step was not performed. Finally, a bit about the MegaRes database. MegaRes incorporates sequences from other ARG databases such as ResFinder, Argonaut, CARD, and NCBI, and is curated and annotated so as not to contain redundancies. It also organizes ARGs in an acyclic, hierarchical way, containing 5 levels. These levels are shown here, with counts as of the 2020 publication and an example entry. At the top is the type of compound resisted, which can be drugs, biocides, metal, or multi-compound. Next is the class of compound resisted, followed by the mechanism by which the gene confers resistance. Next is the gene group, which represents a particular gene or operon. And finally is the individual sequence or accession of the gene, which I will be calling the gene ID. Results. First off is a summary of unique gene ID and gene group counts for the short read and contig MegaRes AMR results. We found 183 and 59 unique gene IDs and gene groups respectively, and in both cases a greater number were found within the short read results. However, we found that there were a number of ARGs unique to either classification source, that is, found within only either the short read results or contig results. Differences in counts could be due to the different alignment methods of AMR++ and Abricate, or the longer source sequences via the contigs. But if we take a look at these proportional Venn diagrams here, we see that the ratio of short read gene IDs to contig gene IDs is far greater than the ratio for the less specific level of gene groups. What we are seeing is likely the loss of strain level granularity during contig assembly. For the contigs, we ran Abricate for ARG detection not only against the MegaRes database, but also against the 4 other ARG databases from which the core of MegaRes was compiled. Here we compare the intersection of unique gene groups across the contig Abricate results and the short read results. The bars show the number of gene groups within the intersection indicated by the bubble lines underneath. Why make this comparison? Well, MegaRes is updated less frequently than its source databases. We see that the short read results still contain far more gene groups than the contig results, and that in total there are 72 unique gene groups found in the samples, 13 greater than when only considering MegaRes results. For a comparison across samples and a look at the types of ARGs detected, here are the relative abundances of compound classes found in the short read analysis results. Each bar represents one sample. The first, most apparent result is the consistently high relative abundance of tetracycline resistance genes across all samples. This is not a surprise, as tetracyclines have a long history of use in apiculture in Ontario, the source location for these samples. The only other compound classes in which resistant ARGs were found consistently across samples were sulfonamides and aminoglycosides, both of which are categories of antibiotics used in disease treatment in apiculture. Other noticeable results are the presence of multi-metal resistance found in 3 samples, as well as one particular sample with several unique compound classes. For a more granular view, here is a heat map of gene groups detected in the contig and short-read MegaRes results. Please pardon the formatting. The arrow indicates the vertical continuation of the plot. Along the left side of the vertical axis are the gene groups, grouped on the right side of the vertical axis by the compound class to which they confer resistance. Each column represents a single sample. On the left is the presence-absence map of ARGs from the contig results, and on the right is the heat map of normalized short-read counts. That is, the normalized number of reads classified to a particular gene group for a particular sample. Here we can see the same types of ARGs common across samples as before: tetracyclines, sulfonamides, and aminoglycosides, but also, at lower abundance, resistance to the category MLS, which is macrolides, lincosamides, and streptogramin drugs. Another result of note is the wide variety of tetracycline resistance genes present. The remainder of the results are mostly sporadic, appearing in one or a few samples. One peculiar pattern is the appearance of a wide number and diversity of ARGs in one sample alone. This includes arsenic resistance, drug and biocide, multi-biocide, multi-drug, multi-metal, and peroxide resistances. Preliminary analyses of metagenome assembly results produced a genome derived exclusively from this sample and with high AMR content. This genome had a relatively high estimated contamination rate of 9.5% and middling completeness estimate of 70%. However, the genome was taxonomically classified within the bacterial genus Laelaudia, which includes members associated with plant pathogens as well as the gut microbiota of plant-eating insects, potentially revealing an avenue of bacterial and/or ARG transmission from the environment. This, however, remains to be further investigated. This concludes my presentation. I'd like to thank my co-authors and researchers from AAFC, Genome Canada and the BCSI team in which the samples originated, and ISCB and ISMB for allowing me to present today. Here are my references for the presentation. If you have any questions, please don't hesitate to email me at the email address found here.